Thank you. I would like to introduce the next speaker and also articulate why Professor Mills has been invited. Because sure, we've been looking a lot at the relationship between the plant, the fruit, and the wine, but it will be too reductive to only think that this is what drives most of the quality. We cannot ignore the impact that the microbiomes and the biodiversity that's seen in microorganism is bringing to the wine typicity. So in the spirit of not leaving aside some other strong factors impacting the vintage, we thought that having some insight from Professor Mills would be very interesting in order to keep the perspectives and see what other parameters are playing their contributions to make the wine taste so specific and so unique in one site. So without any further ado, Professor David Mills.
Thank you so much. It's a pleasure to be here. It's great to see some students that have been through my class and talk a little bit about microbiology. So I'm a professor actually now in the Department of Food Science at UC Davis. For those of you who've been in my class over the past 17 years, I've actually moved over to food science, so I'm no longer teaching that class. It is now, UC Davis is hiring another microbiologist to start working more on yeast. And I moved over to food science to be actually the endowed chair in dairy science. So I work on what I like to think of as two essential beverages, so that's milk and wine, of course. One that's really important early in life, one that's a little bit later. So I'm going to talk about some research, excuse me, some research that we've been doing over the past bunch of years on using next-gen sequencing tools to understand the microbiota, not just of wines, not just of specific fermentations, but actually the whole system, the winery, the vineyard, because these new tools are just incredible in their breadth and our ability to process thousands and thousands of samples. I start with the concept of terroir. I don't think I need to explain it to this particular crowd. I like this cartoon from Wine Folly, which shows different inputs. And I challenge the concept of terroir only in that I like to be able to measure stuff. So in order for us to understand terroir, we want to be able to measure things so we can try to mechanistically understand what are the influences in a regional character in wine. And this has some nice aspects of this where you talks about climate, of course soil, terrain, and tradition. And I'm not really sure how to measure tradition, but we'll have to leave that one blank. But I think there was an idea that I was searching for the whole 17 years that I was in the Department of Viticulture and Enology, and was, is there a microbial terroir? Is there a regionality both to the winery, to the region that would at least add an element? And right now it's speculation that it would add an element to that terroir. We all know that microbes are important in the winemaking process. This is just a simple cartoon that you all are very well aware of, and I just like to point out that we do a lot of things to control microbes, and that might be adding a starter culture or that might be adding SO2. So winemakers, whether they like it or not, manage microbial ecologies. They try to get their arms around that tree, sometimes successfully, sometimes not, as does any cheesemaker or any other artisan of a fermented food. And we know, because of so many years of study of wine, we know a lot of the players involved. We know that there's a lot of microbes that are associated with the grape, particular fungal species, but also a lot of bacterial species that can influence both the grape physiology, as Dario might have talked about earlier today, but also the microbial consortia that actually enters into the winery. We know that there's a variety of non-Saccharomyces yeast that show up both in the flies and in the freshly crushed grapes. And that is a whole nother ecology that occurs early on. We know that Saccharomyces typically takes over these fermentations, if you're lucky, and provides the main bulk of the fermentation. We know that certain spoilage organisms like Brettanomyces come along And at later stages, when in a sense, the food source and the conditions are right, and it has certainly an impact on the wine. And we know that there's other impacts like cork taint that are microbially driven, but in a sense, a residual product from a microbe left in the cork. So many of these things we understand. We understand their influence, and we understand that sometimes they can not make things so good. So we've been wondering, this question of microbial terroir is particularly associated with a winery for a long time. And we always go back to one of the great questions that people like to talk about is, where do the microbes come from that are actually fermenting my wine? Is it coming from the grape? Is it coming from the winery? And I used to tell my class that the idea that you could bring fresh grape juice into a winery and not get it inoculated is like saying you can walk through a car wash and not get wet. It's going to be really hard to do. That doesn't mean that the grapes don't provide a huge bolus of microbes. They do. So it's trying to tease these apart is something that's going to be very hard to understand. But the real influence on the microbial ecology of wine, of course, is the winemaker. And the winemaker makes all sorts of decisions that control that microbial ecology, nudge it in one direction or another, hopefully not this intimately, but they make the right decisions and push the microbial ecology in one direction or another. Now, back in the day when we wanted to study microbial ecology, when I arrived at Davis 18 years ago, the standard technique was plating. And plating gets you nice, beautiful colonies. These are actually yeast colonies this is actually plated Dolce fermentation from up the road. Wonderful yeast colonies. And you can enumerate those colonies and try to get an idea of what was in that original solution. The problem, of course, with plating is that it's inefficient. You can't run 1,000 samples in a day. I can do that now. It's also inaccurate. It plates only what happens to survive onto the plates. So we have a problem with plating schemes is that they're not representative of what's actually there. So if you look at the different tools that a microbial ecologist might have used over the past 15 years, these are some of them. Certainly plating was the most predominant, but it's very slow and you can't do lots. RFLP, or sequencing, in the old days, this is an old version of it. I used to do something called DGGE a long time ago. And that was another slow but non-culture based method that was nice. And that was taken over by quantitative PCR, which came along in the early 2000s and has become pretty common in the food industry now. And is a wonderful way to target a specific organism and enumerate it. It's not necessarily a great thing for doing all organisms because you can't necessarily ID all the organisms it's hitting per se. So what has happened in the last 10 years is next generation sequencing. And next generation sequencing allows us to look at all of the organisms in a sample, and I'll go through some of those methods. Key thing to this methodology has been the advances in DNA sequencing in the last 10 years. And some of these tools have become available during the generation of the data that I'm going to show you. And it's hard to predict where this is going. It's going so fast. DNA sequencing, as you can see here, went through an inflection point in terms of being able to produce a huge amount of sequence, such that it went up 10 to the 11th in 10 years, and it continues to go up. It outpaces our ability to store the information generated by DNA sequencing. We don't have storage capacity big enough, which is ironic because DNA is a information storage molecule. So that's one of the problems those of us who work in this area have, is the computational nature of this is immense. But here's what you can do in a high throughput way now to look at a microbial ecology of X. And when I say X, I mean a whole winery, every single lot of your production, etc. So you collect samples. I have here more than 300. We actually now do no less than 600 per run. It's harder to collect all the samples and get them all together than it is to process the samples and get them to the sequencer. So the logistics of collecting 600 samples is hard. We actually have to mix and match different runs to put them all together. So we have let's imagine 300 samples. We go through a DNA prep so that we know we're cracking open both the yeast and the bacteria. There's a lot of DNA preps out there. Some of them are inefficient, so you have to know what you're doing. And then you go through a PCR step where you're amplifying the ribosomal RNA genes, either from bacteria or from fungal yeast, in 2 separate PCR reactions. And the idea behind that is the PCR primers land on every bug. And so the amplification will come from every bug that's there, theoretically. So that you're going to get a mixed amplicon. That amplicon is going to represent the different organisms that were present in that original sample. And then you just send it off to the sequencer. And they sequence 1 million different sequences, at least. So we get back, per sample, lots and lots of sequences, 1 million sequences per sample, times 600 samples. Or at least 40, 50, 60,000 sequences per sample, times 600 samples. So computationally, that's what we're facing. Because those snippets of DNA sequence are in the ribosomal genes, there are databases set up that we can actually compare each little snippet of DNA to a database and say, well, what bug did that come from? And I won't go into the tools on how you actually do that, but just to say it's an intense bioinformatic process. And it basically spits out a list of organisms that are the likely candidates of what was there. Sometimes it spits out a list of things saying we don't know what's there, and that happens routinely. So we're still finding things and discovering new organisms this way. And we can actually quantify all the fungal and all the bacterial populations this way. So this is a normal output of what we call a microbiome study. This happens to be a swab of the UC Davis winery. And you'll notice on the bottom, there's different pieces of equipment where my student Nick Bokulich actually swabbed that piece of equipment. And then we just crunched up all the DNA that was in the swab and did this amplicon sequencing. This happens to be just the bacteria. But I'm just showing this because it shows you a representation of how this looks. The different colored sections of the bar graph there represent different bacterial orders. So this is a taxonomic level that's pretty high up. It's not genus and species. The orders that were present in that particular swab. You can see how many that were there. So if you think your winery does not have this kind of stuff, you're wrong. It has lots of things in it you're not paying attention to. And you can see the different, how it might be different in the different pieces of equipment. And the orders here are names you guys probably are not used to dealing with. And some of them are actually kind of scary. There's Legionelles in there. So ever heard of Legionnaire's disease? That doesn't mean we have Legionnaire's disease in the UC Davis winery. That just means that order, which happens to harbor one species that is pathogenic, happens to be in there. There's lots of other Legionelles organisms. So this gives us a whole new vista of these facilities that we just have never seen before. Now, you can look at this and try to compare different patterns, but eyeballing it is a pretty low-level statistical tool. So what you do is you composite, in a sense, any one of those bars into a diversity index number. And it essentially represents the number of species or number of genera that are there and the amounts of the different genera that are there. And you're able to come up with a summary number so you can place that on principal coordinate space. And that's what this is. And so you end up with little dots on principal coordinate space so you can compare one of these columns to another column in principal coordinate space just to understand who's similar and who's not. And so you can see all these guys up here are similar and they're different in their bacterial composition than these guys. And that's how you read this. The language of modern microbial ecology now are in these PCOA plots. That's how ecologists look at it. So once we developed these tools, we wanted to ask some questions. One is, can we define a regionality to grape microbiota? Microbiota is a term just meaning the collection of microbes. Grape microbiota present at harvest. So when you crush those grapes, and you now have some must, and you just take that sample and you immediately try to figure out who's all there. But is that different in different regions? Is it different by varietal? And this is a study we did quite a few years ago now with the help of Constellation. We collected 240 musts, 4 different regions. There were 8 wineries involved, and we ran both the bacterial and fungal. And actually, this was such a new project at the time. I started it with John Thorngate at Constellation that we actually had no methods for the fungal at the time that we started it. And luckily, the student I worked with was just a genius and was able to develop those methods at the same time. So let's just look at some of this simple data. You would expect a Chardonnay and a Cab and a Zin to have different grape microbiota. They're different plants. Their physiology is different. You wouldn't expect them to have all the same microbiota. And that's one of the things we saw. This is a confusing slide, but it's one of the ways we present this kind of data. There are some PCOA plots here, down here for, assuming my eyeballs can read that, fungal, and up here for bacterial. And if you just look at the different colors, the Cab, the Zin, and the Chardonnay, you notice that the PCOA points pull away from each other. That suggests that those populations are robustly different. And the same thing with the bacteria for the most part. A little bit more overlap with Cab and Zin. But the beauty of this data is you can drill in and ask the question, because you have the sequence tags of all the things that were there. You can say, well, which microbes are actually driving the differences between these two regions? And that's what the pinwheel plots represent. That's just a phylogenetic representation of everything that's there, colored by what's driving a difference for that particular varietal. So you can see, it's, unfortunately, it kind of goes off the end of the slide here, but you can see Zin has more firmicutes. Firmicutes is a phyla you've probably never heard of, but it includes lactic acid bacteria and bacillus. So there was, for whatever reason, we don't know why, the Zins had more gram-positives, bacillus and lactic acid bacteria. And other organisms were similarly more enriched on other types of varietals. Now, sometimes you would say, well, most of these organisms we don't think have anything to do with wine, but actually we've been able to isolate a lot of these organisms from wine fermentations. So we don't know their contributions to wine. The other thing is some of them, like the one down here, Candida zimplenina, is a yeast that only eats fructose, doesn't consume glucose. And some starter culture companies are considering using it as an adjunct. Well, it seems to be enriched on Zinfandel. I don't know why. It must have something to do with the physiology of Zinfandel, but does that mean that there's a beneficial organism that's contributed there that would otherwise not be contributed to some of these other fermentations? I don't know. Well, what about the original question? Can we separate large regions? If you take all the data and you smush it all together, it's kind of a plate of spaghetti. Maybe you could see some separation in the fungal, but it's really hard to separate this out. But again, this is the beauty of this data. You can start piecing apart and comparing one to another. If you compare, for instance, northern San Joaquin Valley to Napa, then it separates out quite nicely. Those are robustly different microbiotas entering those wineries at those two different regions. And if you apply some other statistical tools, you can prove, in this case, again, it's cut off at the top, but this is a Chardonnay where we were applying some statistical tools to say, are the patterns of organisms truly different in these different regions? And you can see with this kind of mapping, you get some overlap of these lines, but not too much. They sort of stay within their group. And you can apply statistics to that and prove that, it's very different down the valley, down along the coast than up in the valley. Now, this is not a big surprise. These are different weather regions, so you would expect perhaps different microbiota, but nobody's ever shown this before. You can look at which organisms are driving these differences, and again, that's what these two plots are for. So we start to understand why a region perhaps might have a different microbiota every year. If we looked at it year to year, we just did a very small trial in 2012, and the long and the short of the year-to-year variation was that two regions that are this far apart are more different than any one region is year to year. But that's on an N of 1, practically. Now, we also did the same thing with the fungal and showed that there was definitely regionality in the Cab, but not the Chardonnay. You can see the p-value of the Chardonnay there. So this is hypothesis generating. This is not conclusions. This gives us ideas on why things might be different so that we can dig down and actually try to mechanistically understand. How small of a region can we actually differentiate? We can differentiate a really small region. Basically, we can get down to vineyard rows. So these tools are so sophisticated that you can get down to really small regions. So I like to think of the future of where this is going is to start understanding major difference in wine regions. People are doing this for their human gut microbiota now and placing themselves, or their poo, on a map like this, looking at their own gut microbiota of all other humans. Well, we could do the same thing for the wine industry and try to understand year to year, where do you fall relative to everybody else in the microbiota entering in your winery? Can we use that as a discriminating factor for your wine? Is a barcode of this sort going to be an identifier of your specific terroir that year? Of course, to really understand this, we got to get to the influence on sensory, and we've been working on that at UC Davis, and I hope to be talking about that publicly in the near future. I think she gave me the 5-minute signal, so with the 75 slides I have left, that's going to be a problem. So I might skip over a little bit of SO2 and say SO2 affects the wine microbiota. I know that you're really surprised by that. The long and the short of it, it was that the SO2 affected the yeast very little and the lactic acid bacteria tremendously. And in the absence of that, this is actually the yeast profiling with different amounts of SO2, or excuse me, the bacterial profiling with different amounts of SO2, the background microbiota that came in on the grapes was really forward. So Erwinia, a grape-associated organism, was really the predominant bacteria once you add a little bit of SO2. Does that affect the wine? I don't know. Is it dead? We're still trying to figure that out. Remember, anytime you're measuring DNA from pretty much any source, you're measuring both live and dead and everything in between. So we're still trying to understand that. We also looked at wineries, and can we actually understand how microbes move through a winery? So we actually swabbed the whole UC Davis facility a couple years ago, and one of the things we could do is create heat maps of different clades of organisms that showed up in different regions before the season started, right in the middle of the season, and then after everything had been cleaned up. So this is just different heat maps listed with different fungal on the left, bacterial on the right, and I'll just talk about 2 points. Saccharomyces cerevisiae, before the season starts, before a single grape comes into the winery, starts showing up around our garage door and in front of our fermenters. I have no idea why. We're imagining it's coming in on people's boots. There weren't that many flies around at that time. We have no idea. I thought this was a fluke to UC Davis until I tested several wineries in Napa, and the same thing happened. So you guys have Saccharomyces at your door before a grape comes in. But I don't know why. I don't know how it's getting there. Second thing we noticed was that there were some Brettanomyces. This is called Decara bruxellensis, Brettanomyces bruxellensis, in one of the fermenters before the season started. And that didn't make any sense because the winery was clean the previous year, and it didn't make sense at all until I talked to my chair, David Block, who had actually stored his unfiltered wine in that tank from the previous year. And he said, you know, it smelled bready. Now, we got some of the wine and we tried to PCR it out, but we couldn't PCR anything. But if DNA sits long enough in a polyphenolic solution, sometimes it becomes un-PCRable. So I don't know if there was stuff there or not. We just haven't been able to figure it out. But I really wonder if we were seeing the footprints of last year's vintage in that winery as wine moved through. So the last part I want to talk about is just a little bit more about these tools. We actually did we published this in eLife last year, a spontaneous brewery that actually did lambic and regular beer brewing in the same facility, which I don't recommend from a microbial standpoint. And they had different paths through the brewery for doing this. And one of the tools we applied was a really interesting tool. You can take the microbiota of a substrate coming into a facility. So in a brewery, you can imagine grain comes in, hops comes in, yeast comes in, etc. Humans come in. You can do the microbiota of that substrate, and you get a pattern. And then you do the microbiota of the facility, and you get a whole bunch of patterns from the facility, and you just ask, which part of the facility is most like the grain microbiota? Which part of the facility is most like the hops microbiota? And we did that, and that's what this coloring is. You can see different parts of the brewery are differentially colored because they're more similar to the grain, the hops, the yeast, or the beer, and we actually did compare it to human. There was one spot that was more similar to human than anything else. And so these bioinformatic tools are really amazing, and it's moving fast. So being able to do this quickly and source your problems, I think is coming quickly. So in summary, I'd like to say these tools are advancing rapidly. They'll allow complete mapping of fungal and bacterial the continuum all the way through this process. Regional, sub-regional, row by row differences we're able to figure out. Certainly we know varietals are different, and we started to track some data towards that. And I think that examining facilities is where this is really going. And so when I look to the future of these techniques, I look to, and I'm thankful for the Sloan Foundation, because they came up with a cool project called the Microbiology of the Built Environment. The Sloan Foundation tries to fund things that are in between normal funding agencies. So they try to get differential people together. And they try to get microbiota analysis and building analysis. And so we got one of these grants, Roger Bolton, Kyria Bounty Mills, and myself, and we're putting various sensors in wineries that measure all sorts of things very cheaply in a real-time basis. This is a dairy not too far from here, a cheese facility, where we have real-time online measurements that I can pull up on my iPhone. And it tracks all sorts of things, including people movement if we want to. But matching this kind of data to microbiota movement will allow you to understand the environmental changes that happen in your facility that maybe shift the microbiota either in your favor or your disfavor. And it's really one of these things that I think every food facility will soon be doing. So with that, I'll close. We have translated some of this into a company, and if anybody's interested, I'd be happy to talk to them. All of this work was pretty much done by one student, Nick Bokulich. He is going to have 27 papers for his PhD by the time they're all published. It's an insane amount of effort. He's a genius. And I also want to thank all the funding sources, of course. Thank you much.
Thank you. So we have time for one or two questions.
The post-lunch.
is maybe too technical, but do you think we can use the RNA sequencing for understanding the microbiome of grape? Because there are a lot of libraries now about, that they study the effect of abiotic stress on the transcriptome. And I was wondering if we can use those libraries to detect the microbiome and how it evolves under abiotic stresses.
Sure. I mean, there's a whole segment of this. I know Dario might have talked a little bit about this morning, because Dario is leading that kind of effort on the grape side. I sort of stop when the grape comes in, or I start when the grape comes into the winery, and he's before then. There's a lot of research going in this area. As a matter of fact, I'm part of a 5 million euro project in Europe, which actually funds a fair amount of research on wine, that's solely focused on the microbiota from grape to the winery. And so there's lots of people looking at different aspects of grape development in terms of microbiota, and what happens when you throw pesticides on there, and what happens when you throw fungicides on there, and how is my vineyard in this region different than the same varietal in a different region? There's just so many questions to ask. I have to remind everybody, though, this is only correlational data. This is not a cause. This is just, we're finding, I have to remind that, what is it, the crime rate goes up in the summer when more ice cream is eaten. But that doesn't mean ice cream causes more crime. It just means it's summer. And so we have to be careful when we look at these large data sets that are corollary like this. So we want to be appropriately skeptical. She's doing like this. So thank you. Thank you.